All right, so we're going to start with Rebecca Walker. You can come on up. Accounting for isotopic underexpression increases gaseous nitrogen losses from the terrestrial biosphere. So we're going to continue with our terrestrial theme where we ended up this morning and where we'll go for the next few talks. And thanks for your patience.
Okay, awesome, great. As you just said, my name is Rebecca Walker. My collaborators on this work were doctors Ben Holton, Yingping Wang, and Edith Bai. How do I? Sorry.
Yeah, use the mic.
Awesome. Perfect. Great. Okay. So denitrification is the process by which microbes reduce nitrate, removing it from the soils and returning it to the atmosphere. And this process has implications for climate change, environmental pollution, human health. So quantifying this flux is super important, but also super challenging. It's difficult to get direct measurements of this flux due to the high background N2 gas signal. And it's difficult to scale up point measurements due to the high temporal and spatial variability of this process, its hot spot, hot moment characteristics. And so modeling has emerged as a key tool by which we quantify this process. So work coming out of the Holton lab over the last decade has been using the natural abundance of stable nitrogen isotopes in the soil to try and quantify the nitrogen gas flux. So this model uses the equation there on the left, and that F gas term refers to the fraction of nitrogen that's lost via gaseous pathways. So we're really referring to the denitrification flux here. And so to calculate F gas, we use the delta 15N of soil, the delta 15N of nitrogen inputs, the isotope effect of leaching, which is pretty tightly constrained at about 1 per mil, and the isotope effect of denitrification. And an isotope effect just refers to the fact that the molecule containing the heavy isotope reacts a little bit more slowly, and so you get a buildup of this heavy isotope in the substrate, the reaction substrate. Using this model, we calculate a denitrification flux of about 73 teragrams of nitrogen per year, which is equivalent to about 35% of global nitrogen losses. Blah. There we go, okay. However, this model is super sensitive to that isotope effect of denitrification, right? It's like the main term in the denominator there. So just to illustrate that sensitivity, this graph shows isoclines for solutions for that F gas term, and on the y-axis we have a range of possible isotope effects for denitrification that we can use. So if we use 20 per mil, which is the isotope effect for denitrification that we observe in pure culture studies of denitrifying bacteria, we get an estimated flux of 59 teragrams of nitrogen per year, lost via denitrification. If we use, oh God, I'm so sorry, this mouse is like
use
super...
the arrows on the...
Beautiful, thank you, I'm so sorry. Okay, great. If we use an isotope effect of 16 per mil, which accounts for some underexpression of this isotope effect, which we often get in the heterogeneous soil environment, we get a denitrification flux of 73 teragrams. While if we use an isotope effect of 12 per mil, which is commonly observed in the literature, we get a denitrification flux of 105 teragrams per year. So this range from 59 to 105 teragrams is huge and creates a lot of uncertainty in our model. And unfortunately, we don't have a great understanding of what drives this variation, what drives underexpression of the isotope effect. So my work has been taking a stab at trying to address this. So objectives for this study. Can we develop a mechanistic understanding of what drives underexpression of denitrification's isotope effect? And this could be driven by reaction kinetics, so the carbon, the nitrogen, or the C to N ratio of the soil might influence the rate of denitrification and thus the expression of this isotope effect. Or it could be controlled by the physical soil environment, so perhaps soil texture or water-filled pore space is influencing the delivery of reaction substrate to the reaction site. And this is controlling the isotope effect. And then once we have a mechanistic understanding of isotope effect expression, can we incorporate it into our model, our mass balance isotopically constrained model of global nitrogen gas fluxes? To do this, we used meta-analysis to try and quantify variability in the isotope effect expression of denitrification. We used meta-regression, developed a bunch of models, used AIC to select the best model. And then we incorporated this mechanistic model into our existing isotopic model. Great. So let's dive into results. Perfect. So we found evidence for a strong relationship between the soil C to N ratio and isotope effect expression of denitrification. So on the x-axis, we have the carbon to nitrogen ratio of soils, and on the y-axis, we have isotope effect of denitrification. And you can see that there is a significant negative relationship between these variables with an R-squared of 0.6. So what this tells us is that the reaction kinetics of denitrification are important in controlling isotope effect expression, and this is consistent with the broader literature. So as the ratio of carbon to nitrogen increases, nitrate becomes less available and is more locally completely consumed. And when you consume all of your reaction substrate, that isotope effect isn't observed, right? Because there's no even though you have a buildup in the heavy isotope and that nitrate pool, if the nitrate pool is completely consumed, you don't see it. So now that we have this mechanistic understanding, we want to incorporate it into our isotopic model of denitrification. So to do so, first we need to know the carbon to nitrogen ratio of soils globally, and we estimated this at the biome scale. And it's important to note that the carbon to nitrogen ratios we predict globally extend beyond the carbon to nitrogen ratios that were included in our meta-analysis. So we're extrapolating out a relationship beyond what we have data for, so just something to note. Great, okay, so now we want to use this model to model isotope effect of denitrification globally. So in our two maps here, the map on the left, the solid red map, is an isotope effect of 20 per mil globally. And then the map on the right shows our modeled isotope effect for denitrification. So the darker the color, the greater isotope effect expression, and lighter colors indicate underexpression of the isotope effect. And you can see that underexpression is predicted broadly. Across the globe, we generally see underexpression of the isotope effect. And there's this latitudinal pattern where as you go from high to low latitudes, you get greater isotope effect expression. And our average isotope effect globally is only about 11 per mil, which is half of that intrinsic isotope effect. So that's strong evidence for underexpression, which suggests that maybe previous models using isotopes to try to quantify the denitrification flux were underestimating this flux. So to try and get a handle on how much that underestimation or that underexpression is affecting our fluxes, we're going to incorporate this modeled isotope effect into our existing model. Okay, so here we have the fraction of nitrogen lost as a gas for each half-degree by half-degree grid cell. So again, the map on the left is just using a constant isotope effect of 20 per mil, while the map on the right shows our modeled isotope effect. And you can see that the spatial patterns are pretty different. So for temperate and tropical biomes, there is a small but important increase in the predicted fraction of nitrogen lost via gaseous pathways. For desert and high latitude biomes, the maps are pretty divergent, which suggests to me, this inconsistency suggests that perhaps our isotopic models aren't fully accounting for all of the relevant nitrogen cycling processes in these biomes. So perhaps in deserts, ammonia volatilization is really important, and we're not incorporating that fully into our model. Or maybe in high latitudes, leaching of dissolved organic nitrogen is the most important loss pathway, and that's not accurately represented here. So that's something to think about for future work. All right, using our F-gas term, we're going to try and calculate the mass of our denitrification flux. So to do this, our model assumes steady state conditions, so our nitrogen inputs are equal to our nitrogen outputs. There's no buildup of nitrogen in the system. So our nitrogen input maps include symbiotic and asymbiotic nitrogen fixation, as well as nitrogen deposition. So again, just important to note that we're not including fertilizer inputs in this model. And that gives us a total input flux of 208 teragrams of nitrogen per year. So we times this input by our F-gas term to give us the mass of nitrogen lost via denitrification. And that's what we have mapped out here. So again, the cooler colors represent a smaller denitrification flux, and the warmer the color, the larger the denitrification flux. And you can see that the spatial pattern between the two maps is super similar, but in the map on the right, where we've used our modeled isotope effect to account for underexpression, the hot spots of denitrification get hotter. So we're seeing a greater mass of nitrogen lost via denitrification when we account for underexpression in our model. And this increases our estimate from 54 teragrams per year when we use a flat isotope effect globally to 84 teragrams of nitrogen per year. So this is a substantial increase when we account for underexpression of the isotope effect. Okay, so some future research directions. Our carbon to nitrogen ratio relationship captured a lot of the variability and the isotope effect of denitrification, but it wasn't perfect. So more studies on the isotope effect of denitrification would be super helpful to try and develop more certainty when modeling this parameter, especially from high carbon to nitrogen ratio soils or high latitude soils. Additionally, our global delta 15N map was calculated using the data set produced by Joe Crane, published in 2015, of 3,000 soil delta 15N data points. And this data set is awesome, but it's also primarily from the US, Europe, and China. And it would be really great to have more soil delta 15N data points from the tropics, from high latitudes and deserts. And finally, as I mentioned before, figuring out how we can incorporate alternative loss pathways into our model would give us a better ability to model, nitrogen losses from deserts and high latitude systems. Great, so just to reiterate some conclusions, our meta-analysis revealed a strong relationship between the soil carbon to nitrogen ratio and isotope effect expression. And this suggests that reaction kinetics are key in driving the expression of denitrification's isotope effect. Incorporating this mechanistic model into our existing isotope effect our existing nitrogen gas flux model increased estimates of the global denitrification flux. And they increased estimates from 27% of nitrogen losses up to 40% of nitrogen losses. And this has implications for climate change, for pollution, for human health. Additionally, isotope models of denitrification provide an important benchmark against which other estimates of the denitrification flux can be compared, sort of this independent estimate to compare and try and really pinpoint this flux. And with that, I just wanted to say thank you to our funders, funding from the NSF and also the Australian Academy of Sciences. And then a big thanks to the Holton Lab Group, as well as my lab at the University of Minnesota, the Hobby Lab Group, for all of their support on this research. And with any time that's left, I will take questions.
Question for Rebecca?
Yeah, so seasonal variations and hydrologic losses. So we account for leaching in this model just like by incorporating leaching's isotope effect N, and then basically whatever isn't lost via denitrification, we assume is lost as leaching.
In terms of seasonal variations in denitrification rates?
Oh, I see what you're saying.
Depending on hydrologic conditions?
Oh, yeah.
How that might affect your underexpression estimate?
Yeah, so if we have, so that underexpression is really driven by the rate of reaction. So if you have really anaerobic conditions, it would be likely that we have greater underexpression. Yeah, that's definitely part, hopefully incorporated into this model by trying to predict underexpression. But yeah, I think that like our model right now is only driven by reaction kinetics, and trying to figure out how we can incorporate something like water-filled pore space might explain some of that unexplained variability.
All right, thank you so much, Rebecca.
Awesome.
So we'll invite Ron up here. So we're missing some talks. Aaron Delaria, we don't have your talk up there. Is it Mac or PC? Yeah. So that's the only one we're missing for this session.
Missing mine?
No,
She's the only one missing from the session.
From this early afternoon session. Yeah, we're missing a few for the late afternoon. Okay, but Delaria is the only one. Yeah, just have them use arrows.
Just push
use
through
the arrow.
You
Okay.
can-
Skip the mouse on.
All right. So about a year ago, my co-author Jennifer Mills and I started to look into global data sets of the nitrogen isotopes in soil nitrous oxide to see if we could better understand what it tells us about rates of production and consumption of N2O, but also things about the isotope value of the source flux into the atmosphere. So today we're going to give you our present understanding new insights into stable isotopes in soil nitrous oxide. So there's sort of three major goals that people use isotopes in nitrous oxide for. These, and I'm going to go to the answers that will give you to these three questions. The first one is, can isotopes, nitrogen and oxygen in nitrous oxide, be used to help constrain the global atmospheric N2O budget? And so what I'm going to show you today is probably, but the values coming out of soils are probably not what we expected. The second thing, which is very important and very frustrating, is to understand, well, first of all, can we use isotopes to understand whether soil nitrous oxide is coming from nitrification or denitrification? And the results we have today say it'll be probably difficult to explicitly pinpoint one or the other as the source of the nitrous oxide. And then getting at the real ultimate goal of isotope research to see if we can understand rates of production and consumption of nitrous oxide in soil. I'm going to point out that it's probably going to be very difficult, but there are a couple opportunities or pathways that could be used to understand that. The real problem is there's a lot of isotope data being collected, but there's no physical model being used to interpret them or to tie them together with flux chamber measurements. So there's two ways we study nitrous oxide emissions from soils. One is to put a chamber on the surface. The other way is to put wells in the soil and get gas profiles. What they're monitoring is within the soil, there's both highly depth variable trends in production. These black arrows represent production, the red arrows represent consumption, and you get gas profiles that look something like this for nitrous oxide. They're a real mess because of the variable change rates of these competing processes with various depths in soil. The general model that one has to use from a mass balance perspective is one that incorporates net diffusion, rates of production, and rates of consumption. In our model, we're assuming consumption is a first-order reaction in relation to nitrous oxide concentrations in soil. And we've produced both numerical solutions in MATLAB as well as a simple analytic solution. And we're going to use these today to take a look at some of the existing data sets and see what we can learn about them in relation to processes going on in soils globally. Well, one simple implication of solving this mass balance model is that the nitrous oxide concentration or isotope composition at any depth in the soil is a function essentially of three components: nitrous oxide from the atmosphere, the biological production term, and then whatever the consumption effect is on the soil nitrous oxide. So one thing is, if you take nitrous oxide data from soils and plot it as 1 over N2O versus delta N15, this should be and has to be linear if there's no consumption of nitrous oxide. However, if there is consumption, in most cases, this relationship is going to be nonlinear, and also the high N2O samples will not represent the pure biological N2O signal. There's hundreds of data now that have been published on soil nitrous oxide concentrations and isotopes, and I've gathered them all here in one data set: Goldberg and Gebauer, Rock et al., Van Groningen, and Park et al. They're from 5 different parts of the world. Basically, in every one of these soils, the relationship between 1 over N2O and delta N15 is nonlinear. Basically, gross consumption of nitrous oxide is occurring in all soils, even some super dry soils like the sites that were studied by Goldberg and Gebauer. We can show this in modeling space, how this arrives. This is the global data set over here, 1 over N2O versus delta N15 value. This is a tropospheric end member. Over here are some potential biological sources between -40 and -20. But using a model, we can essentially recreate this. Using a model with decreasing production with depth and no consumption, we get a nice linear trend because the N2O versus delta N15 value is a linear relationship. But if we add production and increase the production rate, the curves get more and more nonlinear until eventually the soil flips over to a soil of net consumption, and then the curves go over this way in the other space. And essentially what they start to do is fill in some of these spots on the global soil database. Basically, all these soils are experiencing net consumption at varying rates, and some of them have switched over to totally being a net consumer of nitrous oxide and have isotope values associated with that process. Well, one of the reasons people started studying isotopes and nitrous oxide a decade or more or two ago is to help constrain the role or the amount of soil nitrous oxide fluxed into the atmosphere. And a few years ago, a paper by Snyder et al. Did a new meta-analysis of what the presumed or the average isotope value of the soil flux to the atmosphere is, using flux chamber measurements and then high concentration soil values, which they assumed would be what flows out of the soil to the atmosphere. They calculated that the delta N15 value of the flux is about -17, and the delta 18 value is about 30. We decided to use the diffusion model to explicitly calculate from all these existing soil profile data what the flux is and the value of the flux is to the atmosphere. We found something very, very surprising at first. So this is the data from the Goldberg study in Germany. This is N2O flux, and this is delta 18 value of the flux, delta N15 value of the flux. The green line is the global average from Snyder, and this line is off by 20 per mil. It should be here at -30. But both for oxygen and nitrogen, what we found is as the net flux from soil declined to 0, the delta N15 values became exceedingly negative. And once the soil switched over to being a net consumer of N2O, the values became exceedingly positive. And then as the net consumption increased, they hovered around 0. So this puzzled us at first, and then we found out that both for numerical and analytic models, the same trend occurs. As N2O flux goes down, the delta N15 values go to more and more negative values. Sort of one way to explain this verbally is that the net flux from soil that we measure in a chamber or using diffusion modeling is the sum of two gross fluxes, one from the soil to the atmosphere and the other from the atmosphere to the soil. At low net flux rates, with a consumption term, there's an influx of heavy atmospheric N2O in the reverse direction, and the balance of these two fluxes produces this exceedingly more negative delta N15 value of the net flux. So using in a model, we can tear apart the concentration profile in a soil to the biological component and the atmospheric component when we have consumption. The delta N15 value is essentially the sum of the biological term and the atmospheric term. And the net flux out of the soil in this model run, which the biological flux is -32 per mil, the actual net flux with an overlying atmosphere of tropospheric N2O is -79 per mil. So this is what happens when we approach basically very low production rates. The consumption term gets large relative to the production term, and we get these exceedingly high or low delta N15 values. The question might be, has this trend ever been observed in chamber flux measurements? And I haven't done the most exhaustive analysis yet, but first of all, we have to have low flux rates, and we have to be a natural system so we don't have fertilizer mixing up the signal. So this is all the hundreds of global soil data, again, showing this trend of, on one side of the production, net production here, net consumption here. There's two studies, one from a Brazilian forest by Park et al., and one from Japan by Kato. It does tend to show this. The very low production rates on the positive side get very negative, and then when these soils switch over to net consumption, there's a few very, very high reported delta N15 values. And finally, can we use isotopes to constrain gross rates of production and consumption? And I'll say that the isotope signal is in the soil data, but the problem is the atmospheric boundary condition obscures what that ratio is. And so there's two possible approaches to ferret this out. One is to do the experiments or flux measurements in a chamber that has absolutely zero N2O to remove the tropospheric boundary condition, or the other way is through curve fitting. So this is a model run, but in the field you could probably see something similar to this, where you have an N2O profile that looks like this and it increases with depth. The delta N15 value reaches up to, say, minus 20 per mil in this model run that we did. You can take this model and run it with no tropospheric N2O boundary condition, and then you get the pure biological signal, which would be at the surface, the flux out of the soil is 32.8 per mil. In this particular case, there's a lot of consumption that we've added to it, so we're starting to see this consumption on the isotope value. We simply can't take a sample, though, at a given depth in the soil and subtract the tropospheric condition, the concentration and isotope composition, because by the time that N2O has gotten there, it's already undergone consumption and changed its isotope value. So this is a model example, but here is, as consumption to production ratio increases in the soil, the delta N15 value of the flux out gets more and more negative, as one would expect. But if we assume that N2O that's mixing with the biological term is just pure tropospheric N2, and we just corrected it that way, we'd be correcting it as the consumption-production ratio gets bigger. The correction gets bigger and in the wrong direction. So we'd be fooling ourselves to think that that would be a correct correction to the soil data. I'll skip that because it kind of reiterates what I just said. And finally, the other approach, which Jenny and I talked about, we haven't tried it because it seems so problematic, is basically curve fitting. In a chamber or using soil data, you can calculate or measure the net flux of N2O out of soil. You can measure the delta N15 flux, and then use that using a production, diffusion, consumption model. You can fiddle around with the consumption and production terms and try to come up with a combination that fit our data. So basically, in conclusion, the analysis of soil data around the world shows that all soils we see have evidence of gross consumption. The stable isotope of the flux is way different than I had anticipated, and it shows some very interesting trends with net production rates. And there's probably a consumption-production signal embedded in the soil data, but we have to use some innovative ways to tease that out. Okay, thank you.
Time for one question, Ron? Joy.
There was at least one paper so far that's used lasers in the atmosphere to get a sense of atmospheric N2O immediately above the soil. Have you either looked at that, or to give another angle to the same question, looked at whether the position-specific isotope or results from lasers can shed some light on this question?
Well, whatever is in the right above the soil should reflect the net flux, and so, but I haven't looked at to see what lasers have provided. And then I'll just say my extent of isotopes is just nitrogen and oxygen. I don't do site-specific stuff, so I'm not the person to talk to.
That might be interesting to add in, though. Thank you very much. All right. So our next talk is Emily Stugener. Thank you. You can come on up and join us. And then I just want to double check. Can you see if, before you start it, see if Erin's talk is there? Because if it's not, I think she should go ask them. It's not. Erin, I think it might be worth you going and double checking with the speaker ready room, because your talk's still not there.
All right. I guess this talk will dovetail nicely from the previous one. Hi, I'm Emily, and I'm a PhD student in Joe von Fischer's lab at Colorado State University. Do I just press the space?
Yeah, usually arrows.
Okay. Oh.
On the Mac.
Oh, on the Mac. Okay, gotcha. All right, so the nitrogen cycle is one of nature's most complex behaviors. It's comprised of a variety of diverse processes that are increasingly impacted by human activities. And for me personally, one of the most interesting features of the N cycle is its role in producing the potent greenhouse gas N2O. So as we can see in this figure, many microbial end transformations produce N2O. And this has important implications in our increasingly human-dominated end cycle. So it's well understood that excess N inputs into ecosystems increase microbial N2O emissions. And human activities have really accelerated the amount of N going into ecosystems, which in turn has increased their microbial N2O emissions. However, it's not always clear which specific microbial processes are driving these emissions. So different microbial processes can have multiple different controls, and grappling with all of these factors can limit our ability to predict which processes are generating N2O, which in turn can limit our ability to predict the impacts of different plans for managing N2O emissions. So the work I'll present today examines patterns in N2O production by soils and the underlying processes that give rise to these patterns. All right, so we collected soils from N-disturbed ecosystems, and I'm defining N-disturbed as an ecosystem that has excess N due to human activities. So we collected soils from the Lockvale watershed, a subalpine ecosystem in Rocky Mountain National Park that's impacted by atmospheric N deposition. And we collected soils from the Limited Irrigation Research Farm, a corn agroecosystem in Greeley, Colorado, that's regularly fertilized. All right, and so we took the soils back to the lab and we measured the N2O and we used a laser-based spectroscopic analyzer to measure an N2O concentration, which is those 3 large peaks that you see on the absorption spectra. And then we also measured the concentration of N2O stable isotopomers, which are those 3 much smaller peaks that you see. And as a reminder, an isotopomer is just a molecule with the same number of isotopes, but the position of the isotopes differ. And so we used N2O concentration to calculate N2O production rate, and then we convert the isotopomer concentrations into stable isotope ratios. So we present the abundance of the stable isotopomers using delta notation, but with some additional isotopomer-specific notation. So when there's enrichment in the central nitrogen atom in the N2O molecule, we call that the delta 15N alpha position. And when there's enrichment in the terminal atom, we call that the delta 15N beta position. And we call the average of the delta 15N alpha and beta positions the delta 15N bulk. Now, studies have shown that these distinct isotopic substitutions that we observe in the N2O molecule can be indicative of a particular microbial process that gave rise to that molecule of N2O. And this is important because it means that we can observe these isotopic enrichment patterns in N2O and potentially infer which microbial processes in ecosystems under different conditions gave rise to that N2O. So I'll be talking about how I use that information in the results of my first experiment. So we conducted 2 experiments last summer, and for my first experiment, I used a 15N stable isotope labeling approach to ask the question, which microbial N transformations are responsible for N2O emissions from N-disturbed ecosystems? And we hypothesized that our more nitrate-rich agriculture soils would be dominated by denitrification in pretty much all but the driest soil moisture conditions, whereas our more ammonium-rich subalpine soils would exhibit more variable processes. So we performed inorganic N extractions on our soils prior to incubating them, and we saw that our subalpine soils had quite a bit of ammonium, but not very much nitrate, whereas our agriculture soils didn't have very much ammonium, but had a ton of nitrate. Okay. So before getting into the results of my first experiment, I'd just like to briefly describe our experimental design. So we held our subalpine soils at either 100 or 85% water holding capacity, and we held our agriculture soils at 90 or 50% water holding capacity. And for each soil moisture treatment, soils were amended with either 1,000 atom percent excess enriched ammonium or enriched nitrate. So moving forward through these slides, generally orange colors refer to labeled nitrate derived, and blue colors refer to labeled ammonium derived. All right, so let's take a look at how much N2O is coming out of the different soils. So we see that N2O production rate, here on the x-axis we have our soil treatments, and on the y-axis we have N2O production rate. And we see that production rate did vary by soil treatment. But we can see that within treatments, there was no difference in production rate due to stable isotope labeling. And not too surprisingly, we see that those 90% agriculture soils had greatest N2O production rate, likely due to that abundant nitrate for denitrification. All right, so now let's see where the isotopic enrichment showed up in the emitted N2O. So on the x-axis, we have our soil treatments, and on the y-axis, we have the delta 15N bulk from the emitted N2O. And the stable isotope labeling showed us that all of the soils amended with enriched nitrate, so those orange bars, had a very strong delta 15N bulk signal. Whereas the soils amended with the enriched ammonium, so those blue bars, had a much weaker delta 15N bulk signal. And since microbes use nitrate as substrate for denitrification and ammonium as substrate for nitrification, this indicates that all of the soils were dominated by denitrification and that far fewer nitrifying processes were occurring. For the 50% water holding capacity soils, there's a couple of reasons I'm calling that denitrification, but we do still need to run the stable isotope test to see if perhaps dissimilatory nitrate reduction to ammonium could have been occurring. Okay. So to more specifically break down how much N2O came from either nitrifying processes or denitrification, we multiplied N2O production rate by the percentage of emitted N2O that came from either enriched nitrate or enriched ammonium, and we generated these stacked bar graphs. So the orange fraction of each bar corresponds to denitrification, and the blue fraction of each bar corresponds to nitrification. And we can see that in all cases, far more of the N2O produced was coming from denitrification than from nitrifying processes. So this kind of just illustrates that denitrification appears always dominant, particularly in those subalpine soils where there wasn't even that much nitrate to begin with. It just kind of makes you wonder, what sort of redox hodgepodge are these soils made of? And that's motivated our lab to ask the question, is denitrification always the dominant process? All right, so now I'd like us to look in more detail at the particular isotopomer enrichment signals we saw coming out of the soils and what that shows us. So on our x-axis, we have the soil treatments, and our y-axis, we have the percentage of the emitted enriched N2O that's enriched in the beta position, so that terminal nitrogen. And we can see that in all cases, more than 50% of the emitted enriched N2O was enriched in the beta position rather than in the alpha position. So time doesn't really permit me to get into the details, but we interpret the greater enrichment in the beta position from the soils amended with enriched ammonium, so those blue bars, as indicative of nitrifier denitrification. And moreover, we think the pattern in the beta position enrichment from the soils amended with enriched nitrate, so those orange bars, suggests a denitrifier community with diverse pathways, so just perhaps including both fungal and bacterial denitrifiers. But kind of the bottom line is that N2O isotopomers can reveal subtle and potentially really important details about soil microbial communities and processes that we might have otherwise not been able to observe without using this tool. All right, so now I'd like to transition to discussing the results of our second experiment. For the second experiment, we decided to more closely examine properties of our N-disturbed soils to see how we could potentially couple our understanding of microbial N transformations with the environmental factors that ultimately regulate those processes and their N2O emissions. So we posed the question, which environmental properties will be important predictors of soil N2O emissions? And we hypothesized that soil moisture would be the primary driver of N2O production rate. Wetter soils tend to host more denitrification, and denitrification tends to be leakier with respect to N2O production than nitrification. All right, so in brief, we set up this experiment by holding soils over a soil saturation gradient ranging from 10 to 90% soil saturation. And then for each incubation, we measured N2O production rate. So here on the x-axis, we have percent soil saturation, and on the y-axis, we have N2O production rate, but note that that is on a log transform scale. And each colored dot corresponds to a different soil treatment. So I know there's kind of a lot going on in this figure, but the big thing that stood out to us was that we have these two distinct modes of N2O production, low and high. Now, perhaps not too surprisingly, when soil was less wet, so like below 60% soil saturation, N2O production rate was a lot lower. But then you kind of hit that threshold point at 60% soil saturation, and N2O production rate really increases. And so we were able to fit sigmoidal models to all of this data, and actually for each fit, 80 or more % of the variation in production rate was explained. So again, it might not be too surprising that as the soils got wetter, they produced more N2O, but moving forward, what we think would be really interesting is working to better understand which specific microbial processes are driving these low and high modes of N2O production, and also which environmental properties are potentially impacting these different N2O production processes. So stay tuned. Our lab is currently working on generating process-specific N2O production curves. One thing we can do with the model fits that we generated is we can predict things like maximum and minimum N2O production rate for different soil treatments, but we want to understand what predicts these model parameters. So we conducted a battery of soil chemistry analyses, and then we plotted them against modeled maximum and minimum N2O production rate. So for example, here in this figure, we have soil organic carbon on our x-axis and modeled maximum N2O production rate on our y-axis. And we can see that as soil organic carbon increases, modeled maximum N2O production rate decreases, likely because it's a more strongly reducing environment. And so this is just illustrative of our overall approach to develop predictive relationships between environmental properties and N2O emissions. We'd like to expand this data set to include more soils, get more than 5 data points on that figure. But this is an approach that we can potentially use for predicting the parameter values for a model that can then predict N2O emissions, ideally by specific microbial processes. All right, so some key takeaways. N2O emissions from disturbed soils were dominated by denitrification, and even the driest soils hosted denitrification. That kind of tells us that denitrification appears fairly ubiquitous, and that might be worth following up on. Isotopomers seem like they can be a useful tool for partitioning among N2O generating processes, and it seems like we can learn a lot from these molecules of N2O about which specific processes are occurring in soils. And lastly, we're going to continue working towards improving our abilities to predict N2O emissions from different soil microbial processes, ideally by drawing on information from environmental properties and then coupling that information with N2O isotopomers to partition among processes. There's so many people I'd like to thank for all of their help and support throughout this research, and if time permits, I'd be happy to take questions.
Question over here?
Yeah.
Can you say anything about the absolute rates of nitrification versus denitrification in your soil? Or do you know anything about the relative yields of this process?
Yeah, that's a great question. Not exactly yet, but I'd like to be able to follow up on that. It at least seemed to me from this study, although granted we were working with fairly wet soils, which I think had something to do with denitrification appearing like it was the overpowering rate, like that has higher process rates in the soil than nitrification, but I think that it really just kind of depends on the soil moisture, if that helps answer that. Yeah.
Awesome, thank you so much.
Thank you.
All right, so those of you just walking in the room, we're a talk behind because there was a problem transferring the presentations to our room, so thank you for your patience. I'm going to invite Sydney Clark to come up. She's actually one of my students, and Stu and I work really hard in organizing the session and trying to lump talks together that you guys would all find interesting. We had a withdrawn talk, so we're kind of in our gas emissions section of our session, but we had a withdrawn talk, and after multiple tries to get one of our posters to switch to a talk, I strong-armed one of my students into giving a talk. So Sydney is going to join us, and we're going to switch gears, but stay in Lockville, and then we'll come back to more nitrogen emissions in a few minutes.
Here's the arrows.
Here's the arrows.
Okay.
Cool. Thank you so much for inviting me to talk today. And I just want to say a quick thank you to all of my wonderful collaborators here in Rhode Island, but also in Colorado, as well as the suite of people and organizations and volunteers and funding sources that without this project would not have been possible. So high alpine ecosystem monitoring programs in the Colorado Front Range have noted elevated nitrate concentrations in high alpine lakes. And elevated nutrients can cause impacts such as episodic acidification, shifts in nutrient stoichiometry, and changes in biodiversity. And to give you a sense of what that might look like, this picture on the right is an example of a non-native benthic algal species that has been showing up in some of these high alpine systems, such as Lochvale watershed. And a lot of work is being done to sort of understand what are the drivers that are contributing to those, to shifting this algal regime. So there's been this expectation that recent controls on nitrogen oxides, nitrogen oxide emissions, should have produced a decrease in nitrate concentrations that we're seeing in these high alpine aquatic systems, but because of decreasing national trends. And so let's take a look at some of those trends now. Many of you may be familiar with the National Atmospheric Deposition Program maps that they come out with pretty regularly. Here I'm showing nitrate and ammonium concentrations in 1985 and in 2017. And I won't spend too much time talking about them, but hopefully your eye is drawn to the extreme contrasts that are occurring in the decrease in nitrate in the Midwest and Eastern United States between 1985 and 2017, and the increase in ammonium that we're seeing in the Midwest between those two time periods. And it's already been said today, but that has to do with regulations on NOx emissions and lack thereof on ammonia emissions. But what's going on out west? I would say that the trends are less clear. I've highlighted the NADP western sites in the purple circles, and some work we've been doing has shown that of those 57 western NADP sites, if you take a look at the NADP data from 1993 to 2014, we're seeing that 65% of those sites are showing a significant decrease in nitrate, and 46% of those sites are showing a significant increase in ammonium. And if you take a look at alpine sites alone, of which there are 11 of those 57, all of those alpine sites are showing increasing ammonium, but only a 27% decrease in nitrate. And so it's interesting that there are these differences, that we're seeing these differences in these regional compared to these national trends. And so the question is, what's going on? And so nitrate concentrations in these high alpine lakes remain elevated, and the question is why? And more specifically, what mechanisms might be critical for supplying nitrate downstream in these high alpine lake ecosystems? Discussions in the literature suggest and propose hypotheses such as biological processing in soils, nitrified ammonium deposition, melting glacial features or snowpack, and atmospheric nitrate deposition. And so what I would like to focus on today during this talk is addressing this fourth point and asking the question, is atmospheric nitrogen responsible for this elevated nitrate observed in alpine lakes? Some work done in 2010 by Jasmine Saros et al. Was one of the first studies to sort of point out the importance of glacial features in alpine systems. And she had looked at nitrate concentrations in some alpine lakes that were fed both by seasonal precipitation alone, so snowpack, and lakes that were fed by seasonal precipitation as well as glacial melt, and found that the nitrate concentrations were much higher in lakes that were fed by both glacial features and seasonal precipitation. Some additional work done by Mark Williams et al. In 2006-2007 showed that there's a regime shift in the source waters that dictate the end member contribution seasonally. And so this plot on the right is showing end member contribution in percent on the y-axis and time on the x. And what it's highlighting is that as you shift from a snow-dominated system in early summer, shift to soil water dominated, and then finally base flow or melt conditions from melting cryosphere features. And so this seems to corroborate some of Jasmine's work, showing that you see this shift from a precipitation dominated to a more glacial dominated hydrologic regime late in the summer and into the fall. So in the last talk we heard about Lochvale, and Lochvale is one of two wonderful long-term monitoring programs that have been operating since the 1980s in the Colorado Front Range. Lochvale is pictured on the left. And both of these programs, both of these systems have similar ecosystem characteristics. They contain paternoster lakes, are seasonally fed by snow. They're glaciated and have neat cryosphere features such as snow-fed glaciers and rock glaciers, are at similar elevations above 3,000 meters, impacted by urban emissions to the east, and have National Atmospheric Deposition stations on site. And so these two systems present a really nice opportunity and a rich data set from the past to kind of delve into some of these questions. To give you a sense of my sampling scheme, I went to Colorado in the summers of 2017 and 2018, and my goal was to collect samples that reflected max accumulation conditions in June and baseload conditions in August. And I did this in Niwot and in Lockvale. And I collected various in-members or sample types, including atmospheric deposition, so seasonal snow and bulk wet precipitation, highlighted by the purple stars on the map, or on my schematic. Glacial end-member sample types included ice within the ablation zone, rock glacier inlets, and meltwater, surface water in and outflows, as well as soils, but I will note that I'm not going to be talking about the soil data today. So we can use isotopes as a way to quantify atmospheric nitrate by characterizing the delta 15N, delta 18O, and cap delta 17O. And in our lab, we use the denitrifier method to do that, to reduce nitrate to nitrous oxide, and then we can characterize the mass ratios, which we then present using delta notation. If you're unfamiliar with what cap delta is, I've highlighted the relationship that we use to characterize the cap delta 17O. But essentially, we are quantifying the excess 17O, which is a characteristic signature of atmospheric nitrate due to the interactions with ozone. And so every other source has a cap delta equal to 0. And so on this plot to the right, I have delta 18O on the x-axis and delta 17O on the y. An atmospherically oxidized nitrate source is highlighted by the purple triangle, and the meteoric water is highlighted here in yellow. And then you have the terrestrial fractionation line in black, which all mass-dependent processes such as consumption and denitrification fall along. And so essentially your atmospherically oxidized nitrate falls off of that line and is recording that excess 17O from the interaction with ozone. And then your meteoric water source has no cap delta or a 0 cap delta, and everything in between has some permutation of that. And typical ranges of cap delta are between 20 and 34 per mil, and we can use the cap delta value to then quantify the percent atmospheric deposition present in certain sample types or within a system, and we do that taking an average of our representative atmospheric end member. So let's dive into some data. This plot on the left is showing nitrate concentrations in micromoles per liter on the y-axis, and then time is on the x-axis. And we're really fortunate to have some historical data from 1992, 2012 to 2013, and then data from my 2 sample periods, 2017 and 2018. And those are highlighted by different blue colors, which I'm going to flip through and walk you through. So we're starting with 1992 to 1996. 2012 to 2013, 2017, and 2018. And what I hope you'll notice is that there are consistencies between each of these time periods, where on average you're seeing nitrate concentrations at about 20 micromoles per liter. When you compare that to the glacial features in green, while there's less data, we're seeing a general increase in nitrate as you move later into the season, getting as high as 80 micromoles per liter in August. When you tack on the atmospheric deposition in members, if we start looking at snow in purple, it's on average less than 10 micromoles per liter, and when you compare that to wet bulk deposition in bright pink, it's anywhere between 15 to 20 micromoles per liter. And so we're seeing these elevated levels of nitrate from glacial features in these systems that are increasing seasonally, and that's consistent with what's been seen in the past. And surface water concentrations are consistently elevated at about 20 micromoles per liter throughout the season and between these different time periods. Now if we take a look at some isotope data, this is showing nitrate concentrations on the x-axis, again in micromoles per liter, and then delta 15N, delta 18O, and cap delta 17O as you're moving from left to right. And the end members are represented by the same colored dots. So if we start with delta 15N, what I want to point out is that the atmospheric end members, the snow and the wet bulk precipitation, are much lower compared to the surface waters and the glacial features. And you'll notice that there's a lot of, or there's some overlap between those different sample types, and it might be a little hard to assess what's going on currently, but we can glean a lot more information if we take a look at the oxygen data. So if we look at the delta 18O, the atmospheric deposition is highly elevated compared to the surface waters and the glacial features, and elevated in a way we would expect between 60 and 80 per mil compared to other sample types. If we take a look at the cap delta, it gets even more exciting because cap delta is everywhere, so we're able to measure it in every single sample type we have available for conducting this characterization. And the atmospheric deposition is high in that 20 to 34 per mil range I mentioned earlier, and in the surface water and glacial features, it's much lower. And if you recall, I said that non-atmospheric sources have a cap delta of 0. So the only way, and they're unaffected by mass-dependent processes like consumption and denitrification. So the only way that you can really lower that signal is by diluting it with another source of nitrate. So it's interesting that we're seeing these lower cap delta values in these surface water samples and in the glacial features. I also mentioned that we can use the cap delta to characterize the percent atmospheric deposition and quantify that. So this plot is the same figure that we were looking at in the previous slide, except now I've added a color gradient. I'm showing the percent atmospheric deposition, where 100% is in dark blue, and then the orange and the dark red gets closer to 0%. And what we're seeing is that in the early summer, atmospheric deposition represents approximately 20%. And as you move into the late summer season, it drops to 11%. And this is consistent for the 2 sampling periods that I visited Loch Vale and Niwot in 2017 and 2018. And if you also look, the time periods for 1992 to 96 and 2012 to 2013, we're also seeing decreases that are consistent with those reductions in nitrogen oxide emissions. So this is really interesting because we're seeing, so this data is from 2018, and we're seeing that even at 80 micromoles per liter, we're still seeing a presence of atmospheric deposition. It's only about 11%, but that's pretty interesting considering it's after the snow melt period earlier in the season. Okay, some takeaway messages include atmospheric nitrate is present throughout the summer in all surface water samples, but it only represents 20% early in the summer season and 11% later in the season. While glacial melt increases nitrate concentrations downstream, later in the summer, that cap delta shows that 89% of the pool has to be another source, a non-atmospheric source, that's causing that dilution of the cap delta. And the predominant nitrate source is presumably from biological cycling within soils, snowpack under ice, or nitrification of atmospheric ammonium. And one of our current questions is, could delta 15 of ammonium discern an atmospheric source? And we're trying to work through that now. And some next steps include trying to identify distinctions in the different sample types using water isotopes, characterizing the delta 15 of ammonium, and then also using our isotopic observations as well as some modeling to try and parse out mechanisms versus sources. Thanks for your attention. I'll be happy to take any questions if there's time.
So our red light means, well, there's 1 minute left, so there is 1 minute for questions. Any questions from the audience?
Oh boy.
I was trying to look at the delta 17 versus delta 15 of nitrate relationships in your streams.
Have I tried?
Yeah.
Yes, but I would say there's more work to be done there with trying to understand what is going on.
Because we've done that in France, and we've seen that actually a lot of nitrate comes from the denitrification of ammonium from the snow.
Right. Yeah, one of the things we hope to get at by measuring the ammonium isotopes of our atmospheric end members as well as ammonium in soils, if we can characterize that properly, is to see if we can use modeling and that data to try and figure out if it's all coming from ammonium from the atmosphere. Like, is that really driving that biological source that's presumably causing that dilution?
It's there.
Yeah, thank you.
All right, so our next speaker, thank you, Sydney. Our next speaker is Claudia Wagner-Riddle, who will be talking about the characterization of nitrogen transport, biodrainage, and soil gas emissions from diversified cropping.
Okay, thank you. Okay, so good afternoon. Yes, good afternoon. I'm Claudia Wagner-Riddle, and I think I'll be presenting something a little different from the previous talks. So I'm going to talk about agricultural, environmental, and losses. We've heard a few speakers refer to that. And this is a typical sort of climate conditions for the corn and soybean growing regions of North America. The annual crops that we are mostly interested are soybeans and corn. So the first thing that we see here, that it only gets warm enough sometime in April or May, and that's when the soil is bare and gets fertilized. And then a few months later, we have full ground cover, shown here in corn. Right after, this gets harvested. So again, we have a bare soil where some of the residue decomposes and N is again available from that mineralization. And so for about, depending on where you are, 4 to 6 months or longer, we have an area where we have a lot of water surplus, shown here as a difference between the blue bars, precipitation, and the red bars, evapotranspiration. So our question then is, how can we manage these systems so to minimize losses? And in a way, it's really turning these annual cropping systems more into perennial systems by keeping living roots in the soil in the off-season, increasing the crop diversity, and using cover crops, but also keeping the soil covered and minimizing soil disturbance. So I just wanted to see a show of hands of who has an agricultural background. And you guys know what cover crops are? You can give me some examples? Yes? Okay, so that's what we initiated a project on. So on the top left there, you can see corn with a nice green cover towards the end of the season. So that was a mixture of clover and ryegrass that was seeded when the corn was just a few, like 10 to 20 centimeters high. And our interest then is to compare what we call these perennialized rotation, which with cover crops, and also diversifying the sequence of crops. So this is soybean, wheat, and corn, compared to a poor rotation, which doesn't have any cover crops and has 2 years of soybeans, which is particularly bad because the biomass returned is quite small, and then followed by corn. And we are interested in a range of soil ecosystem services. I'm going to concentrate on the nitrate leaching and mitigation of N2O. Our overall research questions are listed here. We're particularly interested in trade-offs between the soil, water, and air quality when we implement these diverse crop rotations. We have a component of winter warming that I'm going to talk in another talk. And ultimately trying to quantify how beneficial, or if beneficial at all, and that feeds into providing information for farmers so that there is an incentive to adopt these innovative cropping systems. So our long-term we're addressing this in a long-term soil isometer facility in southern Ontario. And I should point out, even though I'm calling it a long-term facility, its intent to be a long-term. It's not yet, because it was created in 2016. But our interest is to see how the dynamics of this management, how it affects the dynamics of these soil ecosystem services. So it is, so in 2016, this is how the site looked like. It was excavated, and these concrete wells were installed. And then we have undisturbed large cores. They're 1 meter square surface area by 1.5 meters in depth. These steel cylinders were placed on top of the soil surface, and then very slowly it was excavated around just about 10 centimeters deep, and then the core was pushed in to avoid compaction, and then again excavating a bit more, pushed in until the full depth was achieved, and then the bottom was cut off. This was transported, and sensors were installed at several depths for a range of measurements such as water content, water potential, temperature, gas concentrations, and soil solution samplers. These are weighing lysimeters, so they're placed on 3 load cells so that we can get evapotranspiration as well as drainage. And then what you see from the top is just the ring, so it's covered with soil all around. And our service wells, where all the all the data loggers are, looks something like this, where we also have drainage tanks that receive the water drained out of the bottom. One of the issues with a facility like this is how do we control the lower boundary? So we want to have it as close to the field conditions as possible. So in this case, we have tensiometers in a field that's free-flowing, and the water potential at the bottom of the lysimeter is controlled to match what's happening in the field. So by water being pumped in or out so that the tension matches the field conditions. So we also have soil gas chambers on each lysimeter that are coupled to a tunable diode laser for both CO2 and N2O. We have 18 lysimeters, each with a chamber. And there's a poster tomorrow that will talk about some tests that we have done on the chambers to ascertain that everything is working properly. So what treatments did we apply to these lysimeters? First of all, we have 2 soil types. So I already mentioned 18 soil lysimeters. So 9 were taken from a sandy loam site and 9 from a silt loam site. These lysimeters were transported to the site that I just showed you pictures of. And then we are managing them with 3 replicates. So 3 of each are receiving a non-diverse rotation, which I'll tell you in a minute what that is, and 6 of them a diverse rotation. Of the 6, 3 are heated, and there is a talk on Wednesday where I'll go over some of the results that we're getting with the winter warming on the diverse rotations. So the non-diverse, I should say both started off with soybeans all treated the same in 2016, June to May of 2017, with the idea to get some baseline data and characterize these lysimeters and see that they're all working properly. In 2017, we continued planting soybeans for the non-diverse system, but planted wheat, followed by a cover crop in the diverse system. So you can see these are planted in soybeans and this is planted in wheat. Later in the summer, the soybeans and the wheat was harvested, and a mixture of cover crops was planted, so trying to increase the diversity of and difference in quality and composition of the residue that gets returned. In 2018, so this year, we followed the poor or the non-diverse rotation with corn, also corn in the diverse rotation with cover crops seeded in at about the 6th leaf stage of corn. So today I will only talk to you about the fall 2017 and winter, well, up to about May 2018 data. But I'll start off with just showing you the data for the baseline year in terms of drainage. So shown here in kilograms. Conveniently, the lysimeters are 1 meter square, so that gives us millimeters. And this is just beginning of 2017, so the winter period, the air temperature shown in green, and the blue envelope of data is for all the loam 9 replicates, and the pink for the sand. So this gives us some confidence that they're all behaving sort of in a similar way, and there are no big outliers in terms of a lysimeter that had a big hole in it that gave us an extreme value. So For then 2017 to 2018, up to June this year, we show here concentrations that were measured every week in the drainage water. And my color code here, green for the cover crop, and black or gray for the non-cover crop, and loam, the darker colors, and the sand, the lighter colors. And it's quite clear that using cover crops reduced the nitrate concentration in the water by a substantial amount, from around 10 to 15 to 2.4 or so milligrams per liter. But we also asked the question, well, how is the drainage amount affected, because that in the end is what affects the amount of nitrate leaching. And what we see is, even though there was a trend for some higher drainage for the cover crop soil, there's not significant difference. But due to those differences in nitrate concentration, we see a reduction in this particular year of between 70 to 77%, which I was quite amazed how large this reduction was. So we see here, again, the black and gray with no cover crops except for some weeds, which of course occur as well, but quite a bit larger biomass due to the cover crop growth here. For the N2O data, I'm showing beginning of 2018. So our air temperatures are quite low for part of the winter. I know that people here in Washington are thinking that they're going through some cold, but I can assure you that it's nothing. But so extended period of freezing conditions with a couple of snow melts. I'm showing in red our micromet N2O flux data measured in an adjacent field. That shows this typical flush that we get at thaw. So the freeze-thaw action induces N2O emissions. And then in green, you can see higher emissions for the loam and the cover crops. So that obviously seems to be a trade-off that is happening, but we see the trend reversing a bit later on in the season, and this is now fertilizer applied to corn. Early on, cover crop a bit higher, but then reversing. So we can't really give you an answer yet on this because we will be looking at the accumulated value of nitrous oxide over the whole year and actually over the whole rotation. So I'm just going to finish with my concluding remarks. We have this long-term site that hopefully will be giving us data for a number of years to come. We're happy with the baseline data. We've seen some significant reductions in nitrate leaching. A trade-off with N2O, and measurements are ongoing. And I'll finish with a pretty picture of our crimson clover and a bumblebee, so increasing other types of diversity as well. Thank you.
Any questions for Claudia? I guess I'll ask one question since N2O fluxes have come up a lot. Do you see evidence for significant consumption in your soils when you have N2O flux drops?
No. Our soils are usually wet, and no, we don't see it. Sorry.
Yeah. No. Interesting. All right. Well, thank you very much.
Okay.
Okay. Our next speaker. So now we're going to switch. We're still talking about nitrogen emissions, but now we're going to switch hemispheres and think about Latin America. So we're going to look at nitrogen emissions in Latin America, impacts, drivers, and policy response. And our speaker is Giselania Cunha Zeri.
How this work here? This, okay. And this one. Okay. So thank you for coming today. Let's turn a little bit to the social dimension of nitrogen aspect. My name is Giselania Cunha Zeri. I work for the Earth System Center in Brazil. And I'm talking today about some results of the Nitrogen Human Environment Network. That was a scientific cooperation network across Latin America countries to investigate the processes that modify different aspects of the nitrogen cycle. We had 6 countries in this project: Argentina, Bolivia, Brazil, Chile, Venezuela, and Mexico. And our biggest problem was that the lack of information on the nitrogen cycle in Latin America is a serious impediment to provide a proper evaluation and projection on how human activity is altering nitrogen pools at the regional scale. Then we developed this conceptual framework of nitrogen emissions in Latin America. The main objective of this framework is to support and integrate a discussion on the nitrogen dynamics in the region, the benefits versus cost situation, impacts, drivers, and vulnerabilities, and also to think a little bit more about policy responses. Because the huge diversity in nitrogen emission sources and pathways requires an integrated approach to deal with these problems. So here we have this conceptual framework. So sources of nitrogen emissions are basically related to productive activities, like agriculture and livestock and other, like industry and transport. These activities, they are conducted by the agents. The agents are the main recipient of the benefits and also the impacts of the uncontrolled emissions. The agents, they are attending a demand for food and energy, which are connected to the socioeconomic factors like demographic trends, economic growth, human development, and many others. Agents are also influenced by the political and institutional factors through the incentives, regulations, and enforcement. These political and institutional factors are related to governance issues, public policies, education programs, and many others. Demand drivers, which is we call the socio-economic factors, and structural drivers, the political and institutional factors, they both influence the agent's behavior, and they also are mutually influenced because they determine the nitrogen emissions balance, if it's balanced or it's exceeding exceeding, its emission it's too high. So also this conceptual framework emphasizes the importance of the political institutional factors in providing and designing the policy responses, which are related to agricultural practices, livestock, soil and land management, nitrogen use efficiency, water use efficiency, emissions control, infrastructure improvement, waste management, and many others that could be turned into policies and directly influence the agents. So they are all connected here. The impacts and risks also feed back the political and socioeconomic factors, and they are all in this kind of connected thing. So when we are doing this, designing this framework, we had so many open questions, so many of them. I just picked some of them here, and I give some insights that we have while we were working on that. And the first one is, what factors are responsible for the increasing of emissions from nitrogen use? And we basically think that production of food and energy are those factors, the main factors. There's many others, and this also depends on the country. For instance, for Brazil and Argentina, the production of food is very important, but for Bolivia is completely different. The amount of emissions from Bolivia is from biomass burning. So they have this difference across the countries. And then the literature shows us there are so many authors affirming that population growth is the main driver of nitrogen emissions. But here we are thinking about so many other factors that are influencing this presumable foremost cause of population, that is population growth. For instance, economic growth, and this is very important, the dietary habits. To illustrate this example, imagine a country that there's a population, and when they have more economical conditions to buy more food and change their dietary habits, the population may remain stable, but the emissions can be high. So it's very difficult to link population growth as the only single factor of the increase of emissions. And finally, what is the role of government institutions in provoking the situation and in controlling and reversing this trend? Some insights that we have, it's about misguided policies that provide wrong incentives to increase production. And also, on the contrary, there's also in the literature that a government with more capacity and there's a good governance is more able to provide mechanisms to curb emissions and to give more control of this situation. Also, we also, the second phase of the work, we performed this survey to search for nitrogen in current Latin American policies. We saw that all countries reported general measures and mechanisms, but in the national communications to the UNFCCC, but only as a greenhouse gas. There are no specific or unified policies dealing directly with nitrogen emissions, but we have found some isolated measures considering the legislation of some countries. This table here shows us some examples. In Chile, Brazil, and Venezuela, there are some laws and policies for specific compounds like NOx, NO3, but they are not connected all in an integrated measure. Why it could be important to connect this? One reason is that, I can say just for Brazil, but federal laws or federal mechanisms, they have some kind of standard for instance, air quality. But on the states, there are different policies, and sometimes these standards are different, higher or lower. And also it's important to integrate because this complexity of nitrogen, it's air, water, soil, so an integrated measure could be much more interesting and effective to deal with this problem. As a conclusion, we can say that Latin American countries are still in the diagnostic phase of the nitrogen problem, assessing and measuring the effects of human-induced changes in nitrogen cycle. Of course, I'm talking about the social-ecological point of view. Demand and structural drivers determine whether nitrogen emissions are balanced or exceeding safe boundaries. Then we do not say that population growth is the driver, but we have two groups of drivers, like demand and structural drivers. Then we have a more broad information, and we can find 4 different causes for different countries. And finally, there's a lack of nitrogen-specific policies in the region, neither a common directive nor a framework in which nations can create their own regulations. I think we have this kind of situation, or trying to do this situation in Europe, but Latin America is not a bloc like a European community, but it should be interesting if you have this kind of framework that could put all the nations together, because we are facing this this problem in a global scale. So I'd like to thank you, especially the Inter-American International for Global Research that was the fund that funded this work, and all the other colleagues that collaborate with that. Thank you very much.
Any questions?
Yes.
Have you had any luck when you talk to policymakers putting the nitrogen problem in terms of services or loss of services? Is there any interest in policymakers in actually acting on nitrogen?
I just can say for Brazil, there is the interest is low, I could say, but there's so many information at the national communication to you and FCCC, but I don't see anything very strong in this way. Yes.
Any other questions?
The task force on greenhouse gas inventories for the framework convention has just had a big meeting on short-term climate forces, including things like ammonia and nitrate, into the reporting requirements. Didn't see several years ago as the process has started, trying to bring up these other compounds and include them in the reporting process.
Yeah, sure. The next national communication from Brazil, they are include more information. They launch it on like a release to general public, and they are trying to connect all this this situation of climate change in like blocks of security, like food security, water security, and so on. And nitrogen must have more importance there, because it's connected to all these things.
Well, thank you very much. Thank you. So our next speaker is Rebecca Abney. I know there's been a lot of changeover with people coming in and out. We're about 10 minutes behind because we had a problem with receiving our afternoon talks on the computers. So we're going to run 10 minutes into the coffee break, but then we will take a break. So Rebecca Abney is going to come up. We're sticking with our nitrogen emissions. Her talk is on soil organic matter and mineralogy as controls on NO2 deposition and HONO emissions from soil surfaces.
All right, hello. Okay, does this work? Okay. Hi, so I'm Rebecca Abney. I'm a postdoc at Indiana University. Which of these keepers? The white one. Okay, and today I'm going to be talking about the relative and comparative roles of soil organic matter and soil mineralogy as they control the conversion of NO2 to HONO. And I want to just start with my acknowledgments to get them out of the way. First, I'd like to acknowledge the members of the RAF Lab who've helped me with conducting this research. The Department of Energy funded this research project, and I worked pretty closely with some people at the Environmental Molecular Sciences Laboratory at PNNL, and I'd like to specifically recognize Sarah Burton, Eric Walter, and Nancy Washington for their help. And then samples and data for this project were also collected from the National Ecological Observatory Network, and so I'd like to acknowledge them for accepting our proposal to collect samples. And just for some brief background for those of you that aren't familiar, NO2, or nitrogen dioxide, is a byproduct of combustion that is a significant source of atmospheric pollution, and it can also react on surfaces to form nitrous acid, or HONO. HONO is another source of atmospheric pollution that can break down to form the hydroxyl radical, which controls the oxidative capacity of the lower troposphere, and it can react to form ozone, which is also harmful to human health. Now this figure on the right here, this shows a vertical profile of HONO, and this panel is the daytime, and then this is nighttime, so the dark dots here is the actual HONO emissions. And you can see that it's more highly concentrated at night near the surface. This is the bottom. or the soil surface. And so soils, this kind of data has been used to implicate soils as a source of HONO, but the specific mechanisms in soil for this production have not yet been fully elucidated. Sorry. So we know that this process is generally formed through reduction of NO2 on soil surfaces and then protonation, which forms HONO. But we haven't fully described this reaction mechanism. And two main sources in soil have been proposed to form this reaction. First, soil organic matter, where quinones and phenols have been implicated in this reaction. And then in iron, you can soil mineralogy, where you have iron reduction. But the relative and comparative roles in soil as to which of these is more important, we haven't determined that. So that is the main aim of this project I'm presenting today. And so first, we're trying to determine which mechanisms form HONO on soil, and then which of these mechanisms are more important in natural soils, and then to try to parameterize these mechanisms so we can use these in chemical and atmospheric transport models. And this is really important because understanding this fundamental chemistry is going to help us represent reactive nitrogen fluxes in these chemical transport models, which are used to predict air pollution and climate change. So to do this, we collected the metadata and soil samples from the Neon Soil Archive. And then we described soil samples, and we used humic and fulvic acids, and we described them using 13C cross-polarization, magic angle spinning, nuclear magnetic resonance spectroscopy, and electron paramagnetic resonance spectroscopy. And these are to describe, we use this to describe the composition of the functional group distribution in soil organic matter. And then we use the EPR spectroscopy to describe some of the iron composition. And then we analyze the neon soils using quantitative X-ray diffraction. And then for each of these soils, we coated them onto tubes and did flow tube type reactions, where the soils were coated onto glass tubes and put into this flow tube reactor. And the flow tube is fitted with a movable injector that can be pushed forward or retracted to either expose the sample or not to NO2 or other air. And then the outflow of this is piped into a chemical ionization mass spectrometer. And we use the SF5- as a reagent ion, which can quantitate HONO. And then, so this was done for all of the experiments using both the neon soils and the humic and fulvic acids. And so for some previous work on using pure minerals in low carbon soils to kind of probe the mineralogical constraints of this conversion of NO2 to HONO, they found positive significant correlations between extractable iron 2 and total HONO flux from the soils. So in this figure panel in the center here, you have pure minerals which were coated onto the tubes and then exposed to NO2, and this is the HONO flux here on the y-axis, and the same thing here on the desert soils. And you can see in the pure minerals, there's a high concentration of extractable iron 2, and then in the desert soils, this concentration is much lower, but the proportional HONO flux is much higher, and this suggests that there's some other component in the actual soils that is responsible for this conversion of NO2 to HONO that we're not yet fully describing, and it's not just the pure minerals. So there's also some previous work that shows the organic matter that's responsible for this conversion from NO2 to HONO. And they found a significant positive relationship between phenols and HONO flux. And this is, so they use, to infer phenols, they used XPS spectroscopy data to kind of describe the surface functional group chemistry. And this is also done with a SIMS-type flow tube reaction experiment. However, this XPS surface analysis isn't very specific, and it could also be aliphatic CO peaks. So to get more specific in this, we describe the soils using soil organic matter 13C NMR techniques, which describe the functional group distributions. And these can reflect differences in vegetation, disturbances, and decomposition in your soil. And so here's the results from the humic and fulvic acids. I don't expect you to digest a lot of this, but the main difference you can see is that the fulvic acids have a much higher concentration of aliphatic compounds, where the humic acids are much more concentrated in organic compounds. And we use these as a model system because they're a widely studied organic matter with a lot of metadata that we can use to correlate with HONO fluxes. And here, so we coated these humic and fulvic acids with kaolinite on these tubes and did similar flow tube reactions as before. And we found a significant positive correlation between the O alkyl peak and total HONO flux. but we didn't find that the phenol peak was a significant correlator with a total honoflux, which was kind of interesting, which suggests that the previous findings of the CO peaks were actually aliphatic CO peaks and not the aromatic phenols. And we also described these humic and fulvic acids using EPR spectroscopy, which we integrated these peaks that we found, and we compared that with an iron standard to find a total iron-3 content in our soils. And we found a wide range of different iron contents throughout the soils, and mostly we found a lower concentration in the fulvic acids compared to the humic acids. But it was good to see that we had a gradient, so we could use that to compare also with a total honoflux. And we found a significant negative correlation with honoflux and iron-3. And this is opposite to the results I presented earlier, where they found a significant positive correlation with iron-2. So this kind of suggests that each of the iron-2 and iron-3 is acting as an indicator of the total redox state of the soils. But we'll definitely need to investigate that further. And so we took these two correlations between iron-3 and the OALQ peaks, the last two correlations I just showed you, and built a multiple regression to kind of predict what we should see in natural soil systems. And when we fitted this data, we found that the OALQ had around a 4-time higher beta coefficient than the iron-3 peak, which suggests that, at least in our model systems, they're more sensitive to soil organic matter than the soil mineralogy. And here we have the same negative trend with iron-3 and the positive trend with OALQ peaks. And so to test this, we selected samples from the Neon Soil Archive. These are all topsoil A horizons, and they're across the continental United States and Puerto Rico. And they span a range of carbon concentration from less than 1% to greater than 12%, and a decently wide range of iron content. And it's a number of different sites from all different types of ecosystems, including temperate forests, deciduous forests, deserts, and prairies. And so we analyzed these soils using 13C NMR and also EPR spectroscopies, and I'm just going to show you the NMR. And here you can see there's a lot of differences in the spectrum, and these are all reflecting the different vegetation types and land uses of each of these sites. And in the future, we plan on actually analyzing a bunch more soils for this project. Yeah, so we took the NMR and EPR and we put them into our multiple regression model to predict the honofluxes for these soil. And of course, they're very highly sensitive to the OALQ NMR peak. And here, so this is the projected outputs from this model where the hono is in these kind of gradient boxes. And then we plotted against total iron and total carbon, and extractable iron, which is in black here. And there's no significant trends here with total iron or total carbon and the honofluxes. And so we think this is indicative that there's other things in the soil besides just total iron and carbon that are driving this. And also, we'd like to further investigate relationships between iron and carbon in soil. So we've already done some preliminary X-ray diffraction analysis of these to describe the mineralogy of these soils. And in the future, we plan to conduct experiments with these soils in flow tube type experiments, and we'll be able to hopefully identify specific minerals that are responsible for some of this conversion. And so here I've described our preliminary attempts to parameterize these atmospheric processes that control the fluxes of HONO and NO2 to and from soils. And the margin between organic and mineral drivers of this conversion is relatively small, and it really needs much more further explanation. And we also need to further understand how the complex interaction between soil organic matter and soil mineralogy can drive this soil reactivity. So I have a couple of quick take-homes. First, that soil mineralogy and soil organic matter each are playing a significant role in this conversion of NO2 to HONO on soil surfaces. And our initial modeling here, it suggests that this conversion may be more sensitive to soil organic matter composition than mineralogy, but we'll be testing this further, so stay posted. And our future research is going to focus on mineral organic matter interactions and testing this across a much wider range of soils. So I'd like to thank you all for your attention. I'd be happy to take any questions.
Questions for Rebecca?
Yeah. Totally. So that's one thing we're going to do in the next experiment. So we have this great collection of soils from the Neon Archive, and our plan is after we do initial experiments, we'll take some of it and do oxalate extractions and maybe some other extractions too if we have time and enough soil. They don't give you more than like 20 grams, so we'll see what we have left. But yeah, and then we'll plan to redo the flow tube type reactivity experiments and see if we can do kind of a subtraction to see what the differential is without the poorly crystalline iron oxides. Yeah.
Well, I was curious what layer of the soil you may have said is thinking of some of these places where there's a thick organic layer and then the mineral soil is quite low. Probably never going to see the M2, so I just wanted to know what part of the soil are you actually.
We've done it with the A horizon. So in the Neon Archive, they kind of like split the soil samples by horizon, and we just kind of went through and picked the top one for all of them. The ones that we looked at didn't have O horizons, but we were trying to like pick the top soil that would be reactive to NO2. Yeah, for sure. We'll have to look into that.
Okay, awesome. Thank you very much.
Thank you.
So our final speaker for this mid-afternoon session is Aaron Delaria, who's going to talk about measurements of leaf-level atmosphere-biosphere exchange of NOx. And then after the coffee break, we'll continue with our final aura session for the afternoon.
Is it this black? Hi, my name is Erin Delaria from the University of California, Berkeley, and I'm going to talk today about the work that I've been doing in Professor Ron Cohen's group on the measurements of leaf-level atmosphere-biosphere exchange of NOx. So our group mainly focuses on understanding the NOx cycle, which is emitted mainly as nitric oxide, NO, from both anthropogenic and biogenic sources. And our goal to understand the NOx cycle has mainly been driven by its role in the production of tropospheric ozone, photochemical smog, and aerosols. But to understand the role of NOx in the production of ozone and aerosols, we don't only need to understand the sources and the chemistry, we also have to understand the sinks. So the main sinks of NOx are by chemistry through reaction to form alkyl nitrates and nitric acid. But NOx can also be removed through deposition of NOx onto surfaces. So the focus of my research has been to understand the deposition of NOx and how the deposition of NOx will affect the chemistry and the global budgets of NOx, specifically the deposition of NO2 to trees. So to understand the potential importance of the deposition of NO2 to trees, we can just look at a satellite image of the Earth and see how much of the total land cover is covered by vegetation. So in a forest, as we've heard a few times today, NOx is mainly emitted as NO from microbial activity in the soils. However, only between 20 to 75% of the soil-emitted NOx ends up making it through the canopy. And this has been frequently modeled in atmospheric models using a canopy reduction factor. One mechanism for this canopy reduction is the uptake of NO2 by trees. The deposition flux can be described by a deposition velocity, the leaf area, and the NOx concentration in the canopy. And observations of this direct uptake of NO2 by leaves has also been observed in many laboratory and chamber experiments. However, some of these laboratory chamber experiments have also sometimes measured NO2 emission at the low NOx concentrations relevant to forested environments, calling into question the role of trees' deposition in this canopy reduction of NOx. So the goal of my research has been to understand the physical processes driving this deposition of NO2 to leaves or possible emission. And our understanding of these physical processes is based on the resistance model, whereby an atmospheric source of NO2 would have to pass through a series of barriers or rate-limiting steps in order to eventually be taken up by the leaf, analogous to resistors in an electrical circuit. The first two resistances are the aerodynamic and diffusive boundary resistance, which are described by the turbulence of airflow above the leaf, the diffusion of NO2 through a boundary layer above the leaf surface, and the microscopic surface properties of the leaf. Once the molecule of NO2 actually gets to the leaf surface, it can either be directly uptake onto the cuticles or the surface of the leaf, known as cuticular resistance, or it can deposit through the stomata of the leaf or the pores in the leaf surface, known as the stomatal resistance, which is equal to the inverse of the stomatal conductance, or the degree of the stomata opening. And then once it enters into the leaf, it can undergo reactions in the mesophyll to eventually become a usable form of nitrogen by the leaf. And these cuticular, stomatal, and mesophilic components of the resistance all contribute to the total leaf resistance, which contributes also with the aerodynamic and boundary resistance to the total resistance to uptake, which is equal to the inverse of the deposition velocity that I mentioned previously. So what I have been doing is taking measurements of the flux of NO2 to leaves and using these measurements of the flux to calculate what the stomatal, cuticular, and mesophilic components are to the total resistance. The way that we do this in the laboratory is using a dynamic chamber setup where we enclose the branch of a tree in a Teflon chamber, well mixed with a fan to minimize the aerodynamic and boundary layer contributions to the resistance. And then we measure the amounts of NO2 that enter and exit the chamber with direct laser-induced fluorescence detection of NO2. And the difference between what we measure at the inlet and outlet to the chamber, we attribute to deposition within the chamber. We also measure water vapor and CO2 exchange to monitor leaf physiological processes. So we've done these experiments so far on a variety of native California tree species. The first was Quercus agrifolia, or the coastal live oak, located along the coastal regions of California. And by plotting the flux of NO2 measured in the chamber versus the NO2 concentrations within the chamber, we get a slope equal to the deposition velocity, or the inverse of the total leaf resistance. And we do these experiments both at the nighttime with the chamber lights off and during the daytime with the chamber lights on. When the lights are off, the stomata are closed, which we confirm with the measurements of water vapor exchange. So the only deposition we see with the lights off is to the cuticles of the leaf. So from these night experiments, we can determine the cuticular contribution to the total resistance. And then during the daytime, we see both deposition to the cuticles and through the stomata. So we can measure the summed stomatal and mesophilic contribution to the total resistance, which we find to be about an order of magnitude lower than the resistance to the cuticle, meaning about an order of magnitude faster deposition through the stomata of the leaf than directly to the surface. We also have not measured any evidence of any emission at low NO2 mixing ratios. We've also done these experiments on Sequoia sempervirens, or the coastal redwood, and this is showing data done at 1 relative humidity. And similar to the oak tree, we can do these experiments with the lights on and off to figure out the cuticular resistance and the summed stomatal and mesophilic resistance. We also find no evidence of any emission, and we find values that are comparable to the oak tree for the cuticular resistance, but about half as fast uptake through the stomata than for the oak tree. For the sequoia tree, we've done these experiments at a variety of relative humidities. In general, when we increase the relative humidity, we see an increase in the stomatal conductance, or the degree of stomatal opening. And we've also done these experiments with 3 different sequoia individuals. And by plotting the deposition velocity versus the stomatal conductance, and fitting this using the resistance model, we can also figure out the mesophilic contribution to this total resistance, which we have found to be about 0.8 seconds per centimeter. So from these experiments, we found that deposition is largely controlled by the stomata, but the mesophyll will become important larger stomatal conductances or stomatal resistances below about 4 seconds per centimeter. In parallel with these laboratory experiments, we've also constructed a multi-box atmosphere-biosphere exchange model where we simulate chemistry, vertical transport, and emission within 7 discrete boxes. And we use this model to assess the impact of deposition of NO2 to leaves on NOx budgets in forests and NOx chemistry. So one set of data that we can look at from these models is the vertical canopy NOx profiles. So this is showing on the x-axis the canopy NOx enhancement throughout the canopy, at 2 different times a day, which we find to be in pretty decent agreement from field observations from our group. We also can look at vertical NOx fluxes above the canopy throughout the day, which we also find to be in pretty good agreement with observations. So one thing that we've done with this model, because we found that the deposition is largely controlled by the stomata, is look at how different parameterizations of stomatal conductance impact the NO2 deposition velocities, NOx vertical profiles, and NOx vertical fluxes. So this first model is showing from Wesley 1989, where stomatal conductance is parameterized just as a function of temperature and solar radiation. So the plot on the upper right shows the deposition velocity of NO2 throughout the day. On the lower left, the NOx vertical profiles at noon, and the lower right, the NOx vertical flux, the day average, with the canopy height denoted by the tree at 10 meters. And we've compared this to a model presented by Emberson et al. In 2000, where not only is stomatal conductance parameterized as a function of temperature and solar radiation, but also as a function of vapor pressure deficit and soil water potential. So this first curve using that model is on a wet day, where it would have a low vapor pressure deficit and a high soil water potential. And this predicts lower NOx mixing ratios through the canopy and decreased vertical fluxes. But this contrasts pretty substantially on a dry day, where trees would get stressed in the afternoon and their stomata would close, causing an increase in the NOx mixing ratios in the canopy and also an increase in vertical fluxes. So a parameterization of stomatal conductance that also uses vapor pressure deficit and soil water potential would be able to account for around a 20% difference in the fraction of soil NOx that ends up being removed by the canopy. And this effect would be pretty important for regions like California that have prolonged periods of drought. We've also used this model to ask the question, where are trees more important sinks of NOx? So this plot on the upper left is showing the fraction of NOx loss that is attributed to deposition, alkyl nitrate formation, and nitric acid as a function of NOx mixing ratio. And in a remote forested region where the mixing ratios of NOx are typically less than 1 part per billion, by far the largest contribution to NOx loss is alkyl nitrate formation, although deposition still makes up around 15%. But in a near urban forest, on the right showing the Berkeley area and the Boston area, which also has a lot of green space near it, and these near urban forests can frequently reach NOx mixing ratios over 10 parts per billion, and under these conditions, the largest sink of NOx is to nitric acid, although deposition would make up over 40% and even more significant at even higher mixing ratios of NOx. So in conclusion, we found that NO2 deposition to trees is mostly stomatally controlled, although the mesophilic control may be important at larger stomatal conductances. NOx stomatal deposition has a large and measurable impact on NOx canopy fluxes. Vapor pressure deficit and soil water potential would be important for accurate parameterizations of NOx, especially in dry regions. And NOx removal in near urban forests may be dominated by deposition under certain conditions. Thank you for listening. I would be happy to take any questions.
Questions for Aaron?
Excuse me, I want to ask, when you test the flux, and you use the dynamic flux chamber or stable flux chamber?
It's a dynamic chamber.
You use the dynamic chamber?
Mm-hmm. The temperature of the leaf in what respect? In the calculation of conductance or just the temperature effects?
In the chamber.
So we do see somewhat of a temperature effect, but we don't have very good control over that in our lab because the air conditioning kind of pops on when the building makes it. But we do see a slight temperature effect, and the trees do get stressed at higher temperatures, but we've tried to minimize that by cooling them down. Yeah, and it mostly affects it by stressing them and having them close their stomata.
Or it looks like you're finding that the uptake is or is entirely first order, it's a linear response. And I was curious if you had any thoughts about why some people
At higher concentrations that it's nonlinear. So with the oak trees, I did some experiments where I went out to 1,000 parts per billion, and I still looked fairly linear to me. So that's what I kind of used when I ran the model that high, and also did those experiments with the sequoia. And so far, it looks pretty linear going out that far. Yeah, I was surprised about that too. I was expecting to be able to see a level off, and then I didn't. So they're all from around the Bay Area, purchased from a local native plant nursery, and they were all from the similar regions, though I forget exactly where, but they're raised in a greenhouse, and yeah.
Do you think there would be any variation across the range within species?
I would be curious if there is. I don't know the answer to that question. I would definitely expect the range between species to be a lot more significant. So that's mostly what we're looking at so far, a difference between conifers and broadleaf species.
Well, I'm going to take a break, but please stick around and Aaron can ask more questions. We have 15 minutes. We'll meet back here to start again at 4 o'clock.
Stage 3.